Sure. Well, first, just as a brief background, there of course remains a significant unmet need for therapies for progressive MS, therapies that are safe, effective, and well tolerated. Vidofludimus calcium is a second generation DHODH inhibitor. It also acts as a NER1 activator. It was found in a phase 2 trial to reduce new lesions in MRI. That was in relapsing MS patients, and is now being evaluated in a phase 2 trial of progressive MS, the CALLIPER trial. That trial was ongoing, but completed enrollment, so is in the on-treatment follow-up period. And so this gives us an opportunity to look at the patients that were enrolled into that. There were 278 patients that were enrolled and compare them to other trials that have already been completed to try to understand the comparability of the patients in CALLIPER compared to those other trials. Now, CALLIPER was open to various forms or phenotypes of progressive MS, enrolled non-active secondary progressive MS, active secondary progressive MS. That was actually a small a subset, and those with primary progressive MS. Now, the CALLIPER trial is ongoing, so only pooled baseline characteristics, including both treatment arms combined together, are available. And this presentation was comparing those patients, those enrolled, to other progressive MS trials, such as Oratorio with ocrelizumab and Hercules with non-relapsing secondary progressive MS and others. And what we found is that the demographics, the age and the sex and things like that were fairly similar between the trials. Primary progressive patients across the trials were mid to later 40s at baseline. That was in the primary progressive trials. Secondary progressive MS patient, though the patients in those trials were also in their later 40s as well. The disease duration across the secondary progressive MS trials were similar, at around 5 to 6 years. But the disease duration was a bit shorter among the vidofludimus calcium patients. Of note, we did find differences in the GAD lesions at baseline and the proportion of patients with GAD lesions. So in the CALLIPER primary progressive trial, 17.8% had GAD lesions at baseline, where the ocrelizumab Oratorio trial had 26.5%. So quite a bit higher proportion of patients with gadolinium-enhancing lesions. Similarly, among the non-active secondary progressive patients in CALLIPER, only 6.8% had GAD lesions at baseline, and that compared to 12.6%, so almost double that in the Hercules tolebrutinib trial. So that lower proportion with GAD lesions, to me, suggests that whatever is eventually seen in the CALLIPER trial is more likely to reflect or to have a higher proportion reflecting the influence of this drug on compartmentalized inflammation, compartmentalized pathology within the central nervous system, compared to what was seen with the other trials, which was a little bit larger proportion of patients with peripherally driven active inflammation.